It's my pleasure to now introduce Dr. Elizabeth Gleim. She is going to be talking about a really important topic. Prescribed fire is a promising tool for reducing the risk of tick-borne diseases. Uh, Dr. Gleim has worked at Centers for Disease Control and Prevention in the Division of Parasitic Diseases. She received her PhD in Wildlife Ecology and Management from the Warnell School of Forest and Natural Resources at UGA. Her doctoral research was based largely at the Southeastern Cooperative Wildlife Disease Study at UGA and the Jones Center at Itchaway in Southwest Georgia. And it focused on the impacts of long-term prescribed fire on tick populations, tick-borne disease risk. She now teaches at Hollins College as an assistant professor of biology. Liz, thank you very much for joining us and talking about this important topic. I will turn the floor over to you.
Awesome. Thanks so much. And thanks everybody for joining me today. I'm going to share my screen here.
Liz, while you're doing that, let me just remind the audience, we will be taking an active Q&A session at the end. So put your questions in the chat or in the Q&A box, and Shan will lead that with Dr. Gleim at the end of her presentation.
Awesome. Yep, looking forward to that. Okay, so as Kevin said, today I'm going to be talking to you all a bit about my research that I've done looking at the impacts of prescribed fire on ticks and tick-borne disease risk. And of course, everywhere I go where I talk about ticks, everyone, of course, has had perhaps the unfortunate experience of having to deal with ticks in one way or another. And I know for some folks may have even experienced a tick-borne disease. So that's just to say that I always know coming into these talks that everyone comes with some experiences and knowledge on ticks. And with that said, there's also sometimes some misperceptions of ticks as well. So I thought it might just be fun to start today with a few fun questions to kind of see what folks know. Shan, I think you might have the poll, I think. But the first question that I had for you all, and we'll launch a poll so you can formally answer, is what do you all think is the most common tick species in Georgia? Is it the black-legged tick, which many people perhaps know as the deer tick, the American dog tick, the Lone Star tick, or the Gulf Coast tick? Shan, are you? Oh, perfect. There it is. Oh, we got quite the menagerie there. So looks pretty evenly split between our black-legged tick, American dog tick, and Lone Star tick. Okay. So it turns out that the answer is actually the Lone Star tick. That is by far the most common tick species in Georgia, and actually the most common tick species throughout most of the eastern United States. They are incredibly aggressive in terms of their seeking out of hosts and are very easily captured when we are out hunting for ticks. Okay, next question I had for you all is the individual with the below rash had what disease? Got a pretty good split there. So majority of folks were thinking Lyme disease. And then we've got approximately a 20-20 split between star eye and Rocky Mountain spotted fever. Yeah, so it turns out that this individual here actually has Southern tick-associated rash illness, which we abbreviate as star eye. And this is a pretty interesting disease that's really only sort of been known for maybe the last 20 years or so. As you can see here, this rash very much mimics the classic bullseye rash that we most commonly associate with Lyme disease. And because of that, we suspect that it was actually probably misdiagnosed for quite a long time before we realized that this was a very different disease that's actually associated with the Lone Star tick, which, as y'all just learned, was the most common tick in Georgia. So if you happen to be in Georgia, as I'm sure most, if not everyone is here, and you develop a rash like this, there's a good chance that it's likely star eye and not Lyme disease. And the good news about star eye, if there's any good news to be had, is that it typically does not have any sort of chronic issues associated with it, as some Lyme cases may have. So very easily treated with antibiotics. Okay, last question for y'all. So generally speaking, how long do ticks have to be attached to transmit the causative agent of Lyme disease? Awesome. So another split, maybe just eking out 24 to 48 hours. And the folks that were thinking that would have been right. For most tick-borne diseases, although not all, the ticks have to be attached for 24 to 48 hours for them to be able to successfully transmit the pathogen. And I say that as a bit of good news in some ways, because really what that means is you've got a decent time window within which you can check yourself and remove the tick. So I always say that the best defense against tick-borne diseases is just getting into a good habit of doing a full tick check each time you come in from working outside. So I know for me during field season, that means every day I'm coming home and doing a check. And if you find a tick, don't wait, remove it as soon as you can. And I'm happy to talk more about things you can do if you happen to find ticks on you to prevent disease, or ways to prevent tick-borne disease or tick bites. But yeah, good to know there are a couple diseases that can be, or pathogens that can be transmitted in closer to 8 hours. But the one that I know most folks are really concerned about is Lyme disease, and that does take a good 24 to 48 hours of attachment. Okay, so with that said, I thought I might give you all just a little bit of basic background on ticks here in the Southeastern United States, and of course in Georgia included in that. So there's 4 different tick species that are of particular human public health relevance. These are ticks that, or species that you will potentially find on yourself if you happen to be outside in the Southeastern US. The Lone Star tick, as I previously mentioned, is the most common tick by far. We also have, of course, the Gulf Coast tick known as Amblyum immaculatum. And this tick is not particularly abundant, but you may encounter it in particular ecosystems, particularly in fairly open pine ecosystems, grasslands, along primarily the coastal regions of the Southeastern United States, or I should say more like the coastal plain, if you will. And then we've also got the American dog tick. And then last but certainly not least, there's a number of different Ixodes species. The best known being, of course, Ixodes scapularis, also known as the black-legged tick, which a lot of people often call the deer tick, although a lot of tick biologists cringe at that name because this tick feeds on many other things besides deer. So we typically use the common name black-legged tick. And what's important to know is that each tick species is capable of vectoring a specific suite of pathogens that can cause diseases in humans. So for example, our Lone Star ticks are capable of vectoring pathogens that can cause ehrlichiosis, as well as that star eye that I mentioned. Our dog ticks, of course, well known for Rocky Mountain spotted fever, and our black-legged ticks, best known for Lyme disease, but of course they have a whole suite of pathogens that they're capable of carrying as well. So over the past several decades, both the incidence of tick-borne diseases, as well as the emergence of new ones, has increased pretty dramatically. And we can see that here with Rocky Mountain spotted fever. We can see that with Lyme disease and so on and so forth. And so one of the big questions is what's driving that? Is it perhaps, better diagnosis, better reporting? To some degree, probably. But most people are in agreement that this is a real trend, that there really are increases in incidences occurring. So the question is why? And really probably at the top of that list is human land modification and land management practices. So as humans are encroaching upon wildlife habitat, that of course is ideal tick habitat. So people are coming into contact with ticks more frequently. I would also argue that probably changes in prescribed fire practices, and in particular, fire suppression in certain parts of the US likely playing a role. And then on top of that, we've got increases in wildlife host abundance. So many of the wildlife hosts that are really important for ticks, such as white-tailed deer, turkeys, coyotes, raccoons, these wildlife populations have really been exploding, particularly in more suburban and urban areas where people commonly are. So as host abundances increase, many times that also means that tick populations are increasing as well. And then the last piece is climate change, particularly up in more northern latitudes. We are seeing certain species, tick species, expanding their range into more northern latitudes than historically they had been able to survive in the past. So as we see this trend, more frequent tick-borne disease, one of the big things that we, of course, are calling for are ways to potentially reduce tick-borne disease risk and control tick populations. And of course, one of those methods that's been proposed is prescribed fire. So there's actually been a lot of different studies that have looked at the impacts of prescribed fire on tick populations. And pretty much most of those studies have agreed that yes, tick populations are reduced immediately after fire. So fire kills ticks. Probably not particularly surprising. And most studies have found that the tick populations will steadily recover over time after that fire. But where we really begin to get differing outcomes is when those studies start looking at sort of the long-term effects of fire on tick abundance. So if we start looking 1 to 2 years out from a fire, are those tick abundances more or less than they were before the fire happened? Some studies have actually found increases in tick populations when we start looking 1 to 2 years out. Others have found sustained decreases. And then others have found that really the populations just return to their pre-burn levels and then just sort of carry on as if the fire had not occurred in some ways. So when I really started getting into this area of research, one of the big questions that we had was, why are there such variable outcomes with these studies? And when we started examining the studies more closely, we saw a couple different things. A lot of those studies in the past had not necessarily accounted for other variables that are known to affect tick populations and tick abundance. For example, wildlife host dynamics, forest structure, microclimate, etc. And or they didn't simulate real-world management practices. And by that, I mean that oftentimes in an effort to be very controlled and scientific, these studies were done on very small areas where maybe the burn was only, say, 1 to 5 acres. And oftentimes it would just be a singular burn done in an area that had not been burned before. And that's in great contrast to what we typically see in most management scenarios, where you're burning dozens, if not hundreds of acres at a time, and you're burning on a regular basis, right? Typically every, say, 2 to 5 years. And those differences have really significant implications for the ability of a tick population to reestablish itself in an area, right? So with that said, what we set out to do was work in southwestern Georgia, and we wanted to determine tick abundance and seasonality, as well as tick-borne pathogen prevalence, and importantly, the impacts of long-term prescribed burning on those different factors. So with that said, we established 21 different sites down in southwestern Georgia. And in order to account for not only what was happening in the site itself, but also in the area immediately surrounding the site, we categorized our sites into 4 different categories, which was areas that were burned areas surrounded by burned areas, so sort of burn utopias, if you will. Then we had burn sites surrounded by unburned areas, and the flip of that, unburned surrounded by burned. And then lastly, we had 3 control sites, which were unburned areas surrounded by unburned areas. And one thing I'll really emphasize here, as I mentioned before, is we wanted to look at the effects of long-term prescribed burning. So what that meant was we really made an effort to identify sites that had been either managed historically with fire or historically had not been burned. So here, when I say burned, I mean that it had been burned for a minimum of 10 years on a regular basis. And if I say unburned, that means that it had been unburned for at least 10 years. And in some instances, they had been burned or unburned for much longer than that. So with that, I set out with myself and a number of different field technicians to commence monthly tick surveys via flagging, which you can see me doing here. We did that on a monthly basis for 2 years straight. Each time we went and collected ticks at sites, we would take microclimate data and weather data. And then on a quarterly basis, we also did vegetation surveys as well as host surveys via trail camera surveys. And I'm guessing you all may be a little curious to know a little bit more about the burns themselves that were happening at these sites. So I basically told the land managers of these sites, just whatever your burn plans are, just carry on. Don't worry about us. We'll just keep on collecting our data while you all do whatever you were planning to do at this location. So ultimately what that meant was these were all dormant season burns. Many of my sites were at the Jones Center at Itchaway, and there they were on a pretty strict 2-year burn cycle. On our state-owned lands, it was more like 2 to 4 years. But ultimately, it all worked out that all of my sites that were technically in the burned category were in fact burned during the course of the study. So by the end of the 2 years of tick collections, we had just over 47,000 ticks that we had collected. The large majority of those were lone star ticks. A distant second were our black-legged ticks. Third most abundant were our Gulf Coast ticks, which was actually somewhat surprising and interesting to us because these ticks generally aren't particularly abundant even in the areas that they do occur. So that was kind of exciting to us. And then bringing up the back were our American dog ticks. So the big money question is, does long-term prescribed fire impact tick populations? The answer is yes. So what you can see here is these are the encounter rates with ticks that were infected with pathogenic bacteria, that is bacteria that causes disease in humans. And what you can see here is in our control sites, our unburned sites, there's normal tick seasonality happening here. And importantly, during peak activity periods, we're encountering about 1 tick an hour that's infected with pathogenic bacteria. And that's in direct contrast to all of our burn sites. So we found that all of our burn sites, regardless of the category that they were in, really behaved the same. And so we lumped them here. And you can see barely any activity at all at these sites, even during peak activity period, peak, I say in quotations, activity periods, very low encounter rates with ticks infected with pathogenic bacteria in many parts of the year, none at all. So this was really exciting to us. And of course, one of the questions we ask when we see reductions in essentially the density of infected ticks is that just because we've reduced tick abundance, or have we also reduced the actual pathogen prevalence within the tick population? And importantly, I'll tell you, tick abundance was significantly reduced at our burn sites. And these were sustained reductions, which was pretty interesting to us because most studies, as I mentioned, would see rebounds of the tick populations over time. We weren't seeing that. They just weren't there at these burn sites. So that was really interesting to us. So then the other big question was, does it affect pathogen prevalence? And that was a pretty unique question because out of all those studies that have looked at prescribed fires and ticks, they've only focused on the impacts on tick abundance. There's only been one study done ever outside of this one that's looked at whether fire could actually impact pathogen prevalence as well. But that doesn't seem like such a crazy idea that it could, because fire, of course, impacts wildlife dynamics. And of course, wildlife hosts are a critical part in pathogen dynamics and pathogen prevalence. So if we're changing wildlife dynamics because of fire, it doesn't seem crazy to think that you might be impacting pathogen dynamics themselves. So with that, we did pathogen testing on all of the ticks that we collected. And what we found was that for almost all bacteria, all pathogens, fire did not seem to have a significant impact on their prevalence, with one exception, and that was Rickettsia, which is what I'm showing you here. And what you can see is that at our control sites, our unburned sites, there was significantly higher prevalences of Rickettsia as compared to our burn sites. And you can ignore the 100% because of course that was a whole 1 out of 1 tick. So this was really interesting to us. One important caveat I'll say is this is Rickettsia within Amblyom americanum, our Lone Star ticks. And in Amblyom americanum, the large majority, and in our case, almost all of the Rickettsia species that we were getting was Rickettsia amblyommatis. And this is actually a non-pathogenic bacteria, meaning that it does not cause disease in humans. There have been a few anecdotal reports, rare reports, where it may opportunistically cause disease in humans, but for the most part, considered to be non-pathogenic. So why do we care if it's not pathogenic? Well, when we looked at all of our pathogenic bacteria, that we didn't see a significant change at our burn versus unburned sites. Those bacteria tend to be carried in the tick population at very low prevalences. Oftentimes, we're talking less than 1 or 2% prevalence, which means that we have a very low number of ticks to begin with that are positive. So if there were any effects of fire on those pathogen dynamics, it'd be very hard to pick up statistically just because the numbers are so low. And so one of our thoughts was that maybe the reason we're seeing significant differences amongst Rickettsia is because it is carried at a slightly higher prevalence. We can see here maybe on average closer to 10% prevalence, which may, of course, just make it or lend itself to reflecting or allowing us to pick up any changes in the pathogen prevalence that we're seeing. So this was exciting to us because in other parts of the country, for example, the Northeastern US and the upper Midwest, where Lyme disease is endemic, the pathogen that causes it, Borrelia burgdorferi, typically is very prevalent in the black-legged tick population. We're talking oftentimes upwards of 40–50% prevalence. And so one of the questions we said is, or thought, was the fact that we're seeing impacts of fire on Rickettsia at least opens the door to the possibility that it may impact pathogen dynamics, particularly in Lyme endemic areas where, again, that pathogen is very common. So another study to do down the line, but certainly something that's quite intriguing to think about. The other thing that we noticed during our study that had not been reported in any previous studies on prescribed fire were very distinct differences in the tick species composition at our burned versus unburned areas. So we can see here in our sort of burn utopias, our burned surrounded by burned areas, that Gulf Coast ticks by far were the most common tick species at those sites. Whereas at our control sites, our unburned sites, the Lone Star ticks, Amblyom americanum, dominated. So this was interesting to us because, of course, with different tick species comes different tick-borne disease risks. And again, this hadn't been reported before. So this was interesting to us. So all this just led to the question of what's driving this? Why are we getting these somewhat unique results in terms of the sustained reductions in tick populations and these sort of unique changes in tick species composition? So to ask that question, we decided to do a manipulative field study. And we had two hypotheses that we wanted to explore. One was that, yes, fire's knocking these tick populations back. But we wondered if what might be actually sustaining those reductions might be the actual habitat itself. So of course, in these systems that were burned on a regular basis, these were pine ecosystems. Many of them were longleaf pine ecosystems, which of course fire is used to maintain those systems, as you all know. And of course, because of that, you have a more semi-open canopy, as you can see here in this photo. You've got a lack of a mid-story, and then of course, a very diverse, typically mostly herbaceous understory. So because of that, you're getting more direct sunlight, you're getting lower humidity, and you're getting higher winds. And what kills ticks? The only thing that can kill a tick naturally is the tick drying out. And so this is a really harsh environment for a tick to try to survive in. So one of our thoughts was just that these ticks just can't survive as well in these really harsh environments. The other hypothesis that we had, which was maybe sounds a little outlandish, but we were wondering if red imported fire ants, which I've abbreviated as RIFA here, so fire ants, if they were playing any role in the regulation of these tick populations. So fire ants are really well adapted to disturbance, and of course, fire is a disturbance. And generally what we saw was that fire ants were quite common in our burned areas, and they in general did not occur in our unburned sites. And believe it or not, there's been a very small number of studies that have actually sort of documented, sort of correlationally anyways, that fire ants may actually predate on ticks. So our question is, okay, is that just sort of anecdotal, occasional predation, or is this really an important driver in tick dynamics in these sites? So to investigate this, we basically set up three different site settings, if you will. The first were burned areas that had fire ants, so that naturally occurs. Then we had unburned areas without fire ants, also naturally occurs. And then we also had burned areas without fire ants. So this is something that we had to create via these fire ant exclusion areas, which is what you're seeing here in this picture. So everything inside of this area here, we would actively work to essentially exterminate the fire ants within that area and maintain the absence of those fire ants for the duration of the study. We would have loved to have had an unburned area with fire ants, but given that they are an invasive species and they don't naturally occur in those settings, we were not about to put invasive species in an area that they didn't already occur in. So we didn't do that, but we did the other three settings. So in each of these three settings, we then established these tick arenas, which you can see here, where I would then place engorged Lone Star nymphs and engorged Gulf Coast nymphs during the times of year in which they would normally be out. And then we waited. We waited and we let them molt into adults, and then we recollected them to see what their overall survival was in these sites. And importantly, I want to mention that in these burned sites, no fire actually occurred during the study period. So we were really just looking at whether the habitat itself was not conducive to survival. So what do we find? Well, first off, we found there's no significant effect of fire ants on either tick species. That said, I will point out, so we've got burned without ants and burned with ants here. And you can see that for Gulf Coast ticks in gray, that they do have slightly higher survival when the ants are absent versus present. And we see that also with Lone Star ticks, slightly higher survival when ants are absent versus present. Now, again, that wasn't significant, but the fact that we're sort of consistently seeing that across tick species implies perhaps they are predating on ticks to a certain degree. Could that be ecologically significant? Perhaps. The other question that we had, or the findings that we had, were that Gulf Coast ticks did survive significantly better in burned habitats than Lone Star ticks. So we can see in both of our burned scenarios, with and without ants, our gray bars, our Gulf Coast ticks, Ammo maculatum, far out surviving our Lone Star ticks in white in both cases. And then we also saw that Lone Stars survive significantly better in the unburned habitat versus the burned. So we can see here in our unburned habitat, the Lone Star ticks here in white doing much better than their counterparts in the burned habitats. Okay, so collectively, what does this all mean? Overall, long-term prescribed fire definitely reduces tick populations, regardless, at least in our case, of burn interval, host abundance, and vegetation structure. We were observing 98% reductions in encounter rates with ticks infected with pathogenic bacteria. So another way to think about that is during peak activity periods, you would only have to spend an hour in an unburned area to come into contact with a tick infected with bacteria that could make you sick. Compared to having to spend probably about 10 hours in a burn site before you would encounter a tick infected with pathogenic bacteria. When we tried to further understand what was driving those sustained reductions, it really seems the key are these vegetation structures that's created by the long-term use of fire. So again, those hotter, drier environments just not conducive to survival. So this really emphasizes the need for long-term consistent burning, of course, which is needed to A, create those habitats, and B, maintain them. And in fact, indicates that singular burns likely would not be particularly effective for long-term tick control, and in some cases may actually be counterproductive. And then the last piece was just, it does sort of beg the question whether or not prescribed fire may actually be a really useful tool for tick control in Lyme endemic areas and other areas of the country. Certainly, given the importance that we're seeing that habitat plays in maintaining those reductions in ticks, begs the question whether prescribed fire would be as effective in other ecosystems and in other climates. So definitely some follow-up work to be done there. And speaking of follow-up work, this is something I've worked on a little more recently, but as we thought about these questions of would this be more effective, or would prescribed fire be effective in Lyme endemic areas and in different climates, also questions about how long it would take to get the sustained reductions that we observed, given that all of our sites had been burned for at least 10 years. And those questions, of course, would take a great amount of time and money to answer. And because of that, models are actually probably a really great way, at least to initially kind of investigate some of those questions. And I was lucky enough to learn about a model that could do that for Lyme disease in particular, which is this LYMESIM 2.0. And this is a model that was developed by a collaborator of mine, Holly Gaff, or I should say updated by her. It was initially developed back in the early 90s, and she more recently updated it just this past year. This is a mechanistic and importantly field-validated model, and it's incredibly complex. I would say it's probably the most comprehensive model on any sort of tick-borne disease system that I've seen, because it does account for wildlife host dynamics, abiotic factors like temperature, the tick's life cycle, as well as the pathogen dynamics that are specifically associated with Lyme disease. And in the past, this particular model has been used to observe how different changes in, for example, host dynamics might affect Lyme disease risk in humans. And it's also been used to investigate the effectiveness of other commonly used tick control methods like spraying of acaricides, host-targeted treatments, for example, that target rodents or white-tailed deer. So I approached Holly and basically said, hey, could we add prescribed fire to this model as another control method that we can investigate, and also compare to these other more commonly used tick control methods like the acaricides that I was mentioning. So we've just done really basic pilot work so far. And so I say take these findings so far with a grain of salt, because we do plan to further refine the model. But what we've initially found, and I'm just going to show you one of the sites that this model allows us to model, which is Cary, New York, so classically Lyme endemic area. And this model quantifies Lyme disease risk based off of DON, that's the density of nymphs, so basically abundance of black-legged tick nymphs, and then the density of infected nymphs, so that is the density or the number of infected black-legged ticks infected with the causative agent of Lyme. And importantly, what you can see is we compared prescribed fire to these other commonly used tick control measures and combinations of those tick control measures. And prescribed fire can be seen here in green, right? And what you're wanting to see is high suppression. Most tick control folks say if you get over 80% suppression, that's pretty good, pretty legitimate. And you can see that prescribed fire really seems to outperform all of the other tick control measures that we've evaluated with this model. Again, I say this with a grain of salt because this is very preliminary and we will further refine the model, but at least initial findings are really exciting and promising. So just real quickly, I just wanted to acknowledge the many folks that have made the work in Georgia possible, the prescribed fire tick work. Of course, CDC, UGA, the Jones Center at HOA, and SCWDS funded the project. My collaborators, Michael Yabsley and Roy Burgess at UGA, Mike Connor at HOA, and Michael Levin and Galina Simpsova at CDC, in addition to many folks at HOA and Georgia DNR who helped support the work. And then for the LYMESIM work, a big shout out to Hollins for funding the project, my student Shravani Chitnani, as well as Holly Gaff and again, Mike Connor. For their assistance with that work. And with that, I will leave you with some fun photos from my work down in South Georgia, and I am happy to take questions.
Awesome talk, Liz. Fantastic. Thanks. You know what? I think Burner Bob needs to change his message, right? Nobody wants ticks in their yard. Right? That's a great reason to do burning. Yeah, we got some questions from the audience here. There were several questions about alpha-gal, which terrifies me because bacon is my favorite food.
Yes.
Do you know what the time frame is for contracting alpha-gal?
So I don't focus a lot on alpha-gal since technically it's an allergy and not like an infectious disease.
Right.
But in terms of alpha-gal, yeah, it's essentially it's associated with the Lone Star tick, and it's specifically some proteins that they secrete in their saliva while they're biting you, which your immune system reacts to. So yeah, and I guess one of the scary things about it is, you know, similar to other allergies, you can develop it over time. So you may have been bitten by Lone Star ticks before and have been fine, but with consistent exposure, sometimes that can lead to development of alpha-gal.
Yeah, but tick checks, right? Daily tick checks, pull those things off. Don't let them be munching on you for a long time and you won't get most of these.
Yeah, definitely for tick-borne diseases, yes. For alpha-gal, probably want to try to prevent tick bites altogether. But yeah. And of course, you know, in terms of preventing tick bites, of course, you know, wear good bug repellent, you know, with DEET, for example. Permethrin treated clothing is a great thing. And, you know, wearing long pants, you know, long sleeve shirts, things most folks are doing anyway that work out in, out in the woods. So, yep.
Sage advice. That's good. Someone was asking you if the tick studies that you mentioned are available to the public. So maybe we could come up with a few links or something where people can do some further reading.
Definitely. Yeah.
And Reese was curious. I'm also curious about, have you looked at growing season burns and if that's more or less effective on these tick populations? And I'm imagining with your vegetation change that might be yes, or is that something you might look into in the future?
That's something I would love to look at in the future. I have not looked at that, and I don't know that I'm aware of any studies that have looked at that. But I certainly think it would make sense that you would probably see some differences between the growing season and dormant burns. I mean, not only because of the impacts it would have on forest structure, but also just because of the tick species and life stages that are active versus inactive at those different times of year. So yeah, I've often thought that would be
Absolutely.
a really fascinating question.
If you need some sites to set up, I've got some great growing season burns in our cache. So we'd
Excellent.
love to host you.
Good to know.
Yeah, anything about burn unit size and distance to unburned areas and how that might affect your data?
Yeah. So when I was classifying the sites as like what was surrounding them, it was literally like adjacent to, so touching, those sites. So one of the questions we had, because we were seeing that even in areas, our sites that were unburned but surrounded by burned areas, we still weren't getting ticks. So clearly there seems to be some sort of indirect impacts on the immediate surrounding area. So one of our questions that we sort of had was, okay, how far did those impacts go? Are we just talking, 50 meters? Are we talking 100 meters or more?
Right.
So that would be a cool follow-up to do. And then in terms of burn unit size, the burn units that I was working in, they varied. I'd have to go back and double check the specific range of sizes that we had. But I would say in general, they were over probably 20 acres at least, but and some were certainly, I would say, probably closer to like 100, 150 acres. What was all I was going to say was, I imagine, particularly if you're in certain parts of the country where they're consistently just doing smaller burn units, that I would imagine the ticks would have a better shot at rebounding in those sorts of places. Yeah.
Cool. Well, we really appreciate your talk, and I guess our take-home message is fire good, ticks bad. I mean, is that a great way to sum it all up?
I'd say that's fair enough. And yeah, I'll be happy. I can maybe send the links to Gina if she wants to the papers that have been published on this, if she wants to share it with folks. I don't know if that's the easiest.
Yeah, if you want to go ahead and email that to me right now, I can post it in the Q&A. And you know what, guys, I did notice that there's not a number popping up in the Q&A. That's where I'm putting people's bios. So just click on your Q&A tab throughout the day so you can see the answers that given to different questions throughout the day. Thanks.
Sounds good. Thanks, Shannon. Thanks, everybody.
Wow, what a great talk. Thank you very much, Liz, and thanks for the wonderful questions. It is now